Good morning, everybody. Can you hear me? Hello? Good morning, everybody. Welcome to the third session called Sensation, Memory, and Action. And I'm Susan Sarah from the Collège de France. I have the honor to chair this session. And I want to thank the organizers for giving me this honor, and especially Nikos for providing this wonderful occasion. And you know, Nikos, just following the tradition of the other speakers, I'm from New York. And I have one foot in New York, and New Yorkers hug. My other foot is in France and Paris, and Parisians, on se donne la bise, but I'm not going to do either one of those right now in the interest of time. So I won't follow the tradition of the other speakers as much as I'd like to. So the first speaker this morning is Anne Grebel, and Anne Grebel is responsible for almost everything we know about the structure and function of the striatum. Over her long career, she has been exquisitely focused on this structure, and since her early days at MIT, she got her PhD at MIT, has joined the faculty there in 1973, and has spent her entire career there. And she is a member of the National Academy of Sciences, the American Academy of Arts and Sciences, and has received many, many awards and honors and prizes for her work on the unraveling of the mysteries of the basal ganglia.
Good morning, everyone. Good morning, Nikos. Good morning, Oksana. I'm going to dedicate my talk, of course, to you, Nikos. Nikos is an amazing friend. Oksana is our friend. Fyodor is not here, but he is our little friend. And I thank you all for being here. What I'd like to talk with you about is this issue, a very fundamental issue in the neurosciences, which is how can we decide to do something and actually do it? It seems like a mystery. We know this is there an echo, ladies and gentlemen? Is the sound okay? Oh, good. So we know that we have to have motivation in order to do this. And we know that if this kind of looping works, in the end, we can think and move. We also know that if this loop is broken somehow, then a number of disorders can occur. The extrapyramidal motor disorders, and then a range of neuropsychiatric disorders which increasingly have become recognized as part of the whole basal ganglia targeting. It now is thought, and what I think the whole field believes, is that somehow these loops that run from the neocortex down to motor pattern generators in the brainstem, up through pathways back to the neocortex, and modulated by dopamine-containing neurons. Somehow this has something powerful to do with how we select actions. But we have to select actions on the basis of evaluating their value, and that brings in a pathway that leads from these prefrontal, medial prefrontal regions that are concerned with mood, motivation, affect, and they turn out to lead down to specialized parts of the striatum. I'll show them to you in a minute. We call them striasomes. And these have the privilege, almost unique privilege, of projecting directly to the dopamine-containing neurons or via the lateral habenula down also to neuromodulators. And so I wanted to show you what these striosomes look like, because I'm going to be talking about them. This is an old slide showing a cross-section through the striatum of a human, and these little pale zones that you see like here and here and here. Those we first identified using cholinergic markers for the cholinergic system, so famous in the field of clinical work. And we soon found that just about all the neurotransmitters, neuroactive substances that were in the striatum were arranged according to this plan. And then I have to show you one other very old finding, which is with Frank Eblin from Germany. We found that of all the regions that we had looked at in red, and they're more on the lateral surface of the brain, here's the genius, corpus callosum, there was this one blue spot which projected predominantly to those striosomes. Now that became more and more interesting because the imaging people found that this same region here in human brain was abnormal in depressed people. Here you can see the brain of a cocaine craver who's in the period of craving. And here you can see, this actually is from Germany, the brain, the same region, roughly the same perigenital region, lighting up in the brains of migrants who have come in but are anxious because they have not lived in an urban environment. So I also thought you might like to see I'm going to have to stop this, which is more powerful than my brain is to do. I'm going to show you an image of a mouse brain. We've labeled, as I'll show you later, the striosomal neurons in red, and you'll see a cut through, a 3D movie, and the nose of the forward part of the brain is up. So here you can see it. I'll blow it up now. And if you look, you'll see that in fact those individual dots, the neurons, are forming what turn out to be labyrinths, taking up quite a large part of the striatum. So we were quite amazed that it is this large. We actually reconstructed it in the human. And so our question then was this. Here we have this evaluative cortex running down to impart the striosomes. I showed you these, but you know, how is this decision brought about? And so how are we going to study decision-making in this context of evaluation? And it's at this point that that Kenichi Yamamori came to the lab. And when Ken came, we decided to use what is commonly used in the clinic, has been for many, many years, an approach avoidance task. And you can get an idea of that here. Here he is, looking your way, deciding whether to approach or avoid you. And here are his friends already running away. They're probably the smarter ones. And it turns out that this conflict approach avoidance decision-making is particularly highly vulnerable in humans in states of anxiety and depression, in fact, used to differentiate them. And so we set up a task, Ken and myself, where we could vary the amount of benefit, vary the amount of cost. I won't go through all the details. And then we could have the monkeys who were looking at combined offers containing both cost and avoid. We could track and mark when they avoided a lot, when they approached a lot, and eventually we could calculate a decision boundary and a difference line. And that was very important to us because we wanted numbers. We wanted numbers because we wanted to be able to microstimulate in different parts of the brain and find out whether that microstimulation, this was all in macaques, caused more approach or more avoidance. And so we also wanted to go after that blue zone here. And so we would be able to microstimulate it, and we did, and this is what we found. We found that relative to stimulation off, the macaques began to avoid more and more. And in fact, we also found that anxiolytics like diazepam used in humans dose-dependently blocked this. There was no effect on our control approach task. And so we were interested to try to go on toward human. And I was lucky to be able to work with people at McLean Hospital in Boston, with the lab of Pizzicato. And here you can see the brain of one subject. Here, do you notice, I hope, this same region is lighting up fMRI as the humans saw the identical stimuli to those used in our monkeys, they're shown here. And this region lit up relative to either the avoidance itself or to the subjective aversiveness of the offer as indicated by the people. So Ken and I went on to ask, could the striatum be involved in this? And so this time I'm showing you just briefly results from our microstimulation in various parts of the striatum. And here again are these cost-benefit plots. Here's the decision boundary when the stimulation is off. Now, and these are summaries. Here the stimulation is on. You might notice there are a few more avoids. And then when the stimulation was turned off, we had a washout period of 200 trials, there was a lot more avoidance. So we looked the next day and we were, well actually we were stunned. So here there is no microstimulation, but here on the next day, the animal was avoiding almost completely. In fact, I hope you can see that over there, it was only by the next day that finally this effect disappeared, and then this time looks about like this time in terms of the decision boundary. And this also was blocked diazepam, and very interesting to us, we found kind of approach sites and avoidance sites, but this prolongation only happened for the negative sites. So we wondered, are we looking at some symbol, some proxy of decision-making, anxiety, depression, because of all the work that had gone on also in the human? So how do we get at that? So we decided we had at least to go to more circuit mechanism. So we changed to mouse, here first the rat. And here you can see the striatum of a mouse. And we've zoomed it out so you can see an individual striosome here. And what we did in our first experiments to look at the circuits, and this is with Alexander Friedman and a number of colleagues, what we did was to set up a very simple two-choice version, kind of cost-benefit approach avoidance test. So if the animal went this way, he got a dim white light, they don't mind that, but diluted chocolate. But on the other end, they get pure chocolate, they love it. But they got a bright light. So what is the choice going to be? And as you might imagine, the little guys mainly avoid the bright light, even though they get a dilute chocolate. And we had actually 4 control tasks. So this is our cost-benefit task. And when we started doing these experiments, we were recording with tetrodes simultaneously in the striatum and the neocortex. We antidromically identified cortical neurons projecting down to the striosomes. And what we found getting echo was a microcircuit leading from prefrontal cortex down to the striosomes, but also to the high-firing internons, those are inhibitory internons in the striatum. So normally, in a normal animal, what we found, as I showed you before, is that animals mainly avoid the bright light. And what we found is when we were in that condition, the normal condition, the striosomes were turned off. But both the neocortex and the inhibitory neurons were turned on. And so what we found was that the inhibition is so fast that it literally cuts off access from the prefrontal cortex to the striosomes. And here I emphasize that the dynamics totally critical. So now what happened was that we manipulated to slow down this pathway, and we turned on the striosomes, these turned off, and we were again stunned because now these animals approached the formerly aversive end. So here they are running to the bad end, and when they do that, the striosomes are blazing out with actually quite high activity. So if striosomes are on, mice approach an aversive stimulus, and if they're off, mice avoid bright light. Now, having worked on striosomes for a while, I was a bit disappointed that it looks like striosomes are bad, but we went on. And what we asked Nikos, I cannot tell you how sorry, there was an airplane mix-up, and I did not hear your talk on state. But what we figured was, could this be a state change? Could it be that the striosomes were a kind of mediating state? They were switched. And so we wondered, how can we study this? How do we get a state that we can really get our hands on in a relatively straightforward task? In other words, could experience alone do this switch? And so here's what we did. We decided that we would use mild chronic stress of the animals. So for 2 weeks, they were either for 1 week put in a little cloth holder. I always think of that as like sitting in the middle seat of like going from Germany to the United States. Or I said an airplane mix-up, United States to Germany. And here I'm showing you then, or we had mild foot shock for 2 weeks, 1 hour a day for either one for 2 weeks. So here's the control. They're avoiding the bright light. Striosomes are off. Now, mind you, we've not touched the brain. And with stress exposure, this is what happened. We didn't touch the brain. They had 2 weeks of mild chronic stress. First of all, the striosomes were on. That was a phenocopy of when we had manipulated this input. But it was really shocking that now the animals approached the aversive light. All we did was to give them mild chronic stress for 2 weeks. This we've done in many, many, many animals. So that began to make us believe that experience could switch the dynamics, and I hope this is somewhat like what you talked about state, Nikos. And so we wondered if this is true, we ought to be able to reverse the effects of stress. Of course, that's a tall order. But given what I told you, you can maybe guess what we did. Here is, first of all, the stressed animal. He's going to the aversive side. And now we have simply changed this circuit by going after the interneurons, and their behavior flipped immediately. They avoid the aversive side. So From this, we're getting the idea that there certainly is an EI balance going on, that a microcircuit in the striatum is exceptionally important for this. It somehow wires up to the striosomes. It's capable of being influenced by the neocortex, and that if we can restore that dynamic, then we can get rid of the effects of stress. Now, I mentioned how we're doing. Oh, we're doing well. In fact, I may have gone too fast. I think I mentioned at the beginning that a special characteristic of striosomes is that they actually can project down directly to the dopamine-containing neurons of the substantia nigra pars compacta. I'm about to show you that. And then they have this ancillary path. We've been working hard on that, but there's no time. And so we wanted to see this in more detail and then to find out what was going on. And so we used birth-dated mice to visualize this input from striosomes down to the substantia nigra. You can see it here. And these white fibers show you the striosomal fibers, which crawl up bundles of ventrally descending dendrites, which are well known inside the field. And here you can see that with expansion microscopy coming, that beautiful, beautiful way the striosomal fibers come up to innervate the dendrites and some of the cells that are containing dopamine. And if we go to expansion farther, we can see this beautiful image. The roses at the top are the dopamine-containing cells. Their dendrites are coming down. They bundle around each other, so we call them dendrons. And here in the green, you can see striosomal fibers that have come all the way down from the striatum, crawling up to tie themselves, entwine themselves with the dendrites, and just reach the cell bodies that contain dopamine. So that raised the question of whether the actual activation of the dopamine-containing neurons could be switched by the fact that neurons coming down toward them themselves are switching on, depending on whether the neocortex is switching on and off and influencing them via that dynamic microcircuit. So this also led to the question, after all, we had seen some effect of experience on the system, could it have to do with learning, with experience-dependent learning? And so we decided to look at this in two ways, and I'll show you that briefly. First of all, we've been doing two-photon imaging. It's really great to see the little neurons, and we were very lucky. We had, I guess I've left out the slides, but we engineered a bunch of mice, and among others, we birth-dated by tamoxifen injection neurons born early, those are mainly striosomes, or neurons that are born later in embryonic life, those are mainly going to be matrix neurons. And through that, I'm sorry not to show you the slides, but through that, we actually could see in the grown-ups not only the cell bodies of these striosomal neurons, these are born maybe E11.5 in a mouse, 10 to 11.5, but the wonderful thing is we could see their processes. And what this meant is that we could identify these clumps, the parts of these labyrinths, which meant that we could then see what happened when the animal ran tasks. And so we saw calcium signals when we did the two-photon, and then what we did was to run a very simple reward-based task in the animals, and you're seeing actually quite similar curves for cue onset, reward onset, very simple task for striosomes and matrix. And then over learning, here we get to criterion. You can see the striosomes are beginning to fire more. And then finally, during the Q period, their activity has grown a lot. The activity of the matrix neurons hasn't. This was our very first hint that indeed, if we image individual neurons, we can see a learning-related change in calcium imaging. So then we've gone on to use not only tetrodes, but photometry while we do slightly more complicated, but still simple tasks, wherein the mice can receive one of two tones. This one means reward is coming. This one means cost is coming. And then here, what they need to do is to learn that for low amount of licks, medium, high, they get more and more reward. But if they refrain from licking, then the more they refrain, the more, the less they refrain, the more the cost. And so the outcome again is good juice. The outcome for the bad side again is light. And so what we found so far is that if we look at the lick frequency, in the rewarded case, the licks, of course, go up, 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 up. And for the cost Q, they sort of stay stable, and by 1,000 trials, they're gone. And just to show you that we can analyze, we've got a lot of algorithms to be sure to analyze these transients well. Here's the control. So those are calcium transients. And so I want to show you one example because it's kind of fun. We're going to look at lick frequency over many, many, many trials. It's a one neuron. So reward Q is in blue, cost Q is in red. So you can see that by the end, there's a lot of blue reward lick frequency and here much lower cost. But what we did was to make line plots here over time showing the Z-scores of the fluorescence. And we had onset reward and then the outcome period. And so we'll look at the outcome period where we saw quite dramatic results. This is showing early on here. And now here is a trial in which the reward was getting a lot of licks. You can see that now this blue signal has become very high. And by the end, here's what we saw. There was a strong, strong signal of fluorescence for the reward one. And this now is the crux of where we stand. Now what you're looking at is the photometric signal, reward minus cost in the response period, versus a discrimination index, which we calculated. You're looking at striosomes across the learning, and what you can see is, indeed, they learned. And as they learned, the signal went up. And if you look at the relation between the photometric signal and a lick response, you can see we really didn't get much of anything for the striosomes. However, if we now look at the matrix signal, now we're using so-called matrix mice that have been tamoxifen labeled late in embryonic period as opposed to what I just showed you for the striosome mice. Now you can see there's almost no relation between the photometric signal and the learned discrimination, but it goes very, very well with the licking, with whatever movement we're showing. So what I've shown you here is that there really is a very dramatic and dynamic system which is flowing down from evaluative cortex down to the dopamine-containing neurons. And we're looking very hard at them now. And we also have been looking to see if we can get at that microcircuit. And so let me end by showing you we now have been able to use an extremely high-resolution microscope. And when we do that, we actually can count. We have a lot of algorithms for automatically and then hand-checking counts of here putative parvalbumin-containing inhibitory contacts. And these are our earliest results. These are a bar graph of the number of putative inhibitory terminals for animals who've learned, and here for animals who have not learned. By not wanting to go over time and to give you plenty of time for questions, I won't show you, I left them out, the initial results of our DREAD experiments, but I might be able to find them somewhere in here. But the initial results are very promising, that we can definitely modulate up or down these signals by using excitatory inhibitory DREADs, and we've done endless controls for CNO, really endless, thanks to the computational ability of Alexander and about, I know you're not going to believe it, 20 MIT undergraduates, each one of whom is roughly speaking a genius. So I'm very, very lucky. So we are interested to go on with this work. I told you about the DREAD work, and there are many, many things we can do. But one thing that's become of great interest to us, and we're finding more and more, is the following. It turns out, and actually it was well known for a number of years, that opioid receptors, especially the mu-type opioid receptors, are enriched in striosomes. That was one early marker and still a marker used for seeing striosomes, the so-called MOR1. But we found in our lab, as was found many years ago in human, but not found in mouse, but now found in mouse in our lab, is that one of the forms of 5-HT receptors is stunningly enriched in striosomes relative to matrix. And then we very recently have found and published that cannabinoid receptors, CB1 receptors, and those are the main receptors for drugs like marijuana, that these, actually those striosome dendron bouquets are encrusted with CB1 receptors, which are also heightened in striosomes. So in summary, what I'm trying to tell you is that unbeknownst to all of us, thinking that the striatum and the basal ganglia were principally related to movement, to Parkinson's, Huntington's, many, many dystonias, but Parkinson himself had noted mood changes, and then other people had, bit by bit, in Parkinson's disease and other disorders. In fact, in Huntington's disorder community, it now is recognized, unlike even 5 years ago, that mood disorders can occur maybe 10 years before manifest, so-called manifest Huntington's disease, when movement abnormalities begin to be seen. So it's of great interest to us that the striosomes have been found to be differentially affected in mood-disordered Huntington's patients. That's work I was lucky enough to do with Richard Fall of New Zealand, and in our own work and the work of others in the case of Parkinson's disease patients with levodopa-induced dyskinesias. Again, the striosomes are strikingly affected, as are genes that we've discovered that I don't have time to talk about. So we're thinking it could possibly be that there is a convergence. As I said at the beginning, striosomes are highly, highly neurochemically specialized relative to the matrix. That's why they were originally found. They're found for almost every neuroactive substance, as I said, that anyone has found. And so our hope is that if many people can continue to work on this system, that we may somehow, all of us in the field, converge on helping to address these terrible problems that humans face. And I would like to thank especially members of our team. Ken and Satoko have just returned to Kyoto. Here's Alexander with friends. Bernard Bloom is the 2-photon here, made some of the mice I couldn't talk about. Here are our collaborators. I learned the tamoxifen labeling technique from Sean. Here at Boyden, here help with 2-photon, and here help with the cannabis. I want to thank you very much for listening.
Thank you, Anne, for this very, very elegant presentation. And now we have plenty of time for questions.
That was a beautiful talk. Thank you very much. I can really appreciate the specificity that you have with primates and with rodents as a model system. And I was intrigued by the human imaging data that you showed, where it was, my question was whether that was striatum or anterior cingulate, if there's some differences.
Not clear. Here's what I meant. In the monkey, we had found this perigenual, pregenual, medial, prefrontal cortex projected preferential striations, okay? And then as I showed you, people with depression, people in cocaine craving and so on, the people studied here in Germany also showed this region as this perigenual cortical region as abnormal in people with these problems. And so what we thought would be a wonderful experiment would be to take everything we could from our monkey experiments, literally, including the cues that we used for the monkeys, including everything we possibly do. And then with the McLean people, have humans do this, okay, test, and then do fMRI. In fact, we've now begun to study people with depression, and I hope soon we can look at people who are suffering from anxiety. So in that image I showed you, it was that perigenual region that lit up just as in depressed people, in the craving people, in the migrants, and in our monkeys. Thank you.
Thank you, Anne. I cannot resist to ask, when you have this beautiful paradigm, when mice change decision preferences, do all of them change or some don't? All mice, all individuals? I'm asking about individual differences and also sex differences.
I tell you, it's absolutely amazing. We did, I wish I could remember the numbers, but it's a fabulous number, 20, 30, 40, 50, I mean, if you combine the mice and the rats. I think every single one, maybe but one, showed this reaction. I told myself I wouldn't go over time no matter what. So the slides are missing, but it was quite stunning. This is something that they do, and my own fantasy is that this is because the pathways I'm talking about are apparently evolutionarily very, very highly conserved, way on back, okay? And also the behavior is critical for survival. If you don't learn whether you, my dear friend, are someone to approach or to avoid, bad things can happen to you, or wonderful things. So I think, I personally am very interested in evolution in the brain and rituals in the brain, many of which I think are conserved. And so that's my guess, but I really was stunned. In the case of monkeys, of course, we had 2, but now we have repeated that in 2 more, and 1 more is ongoing.
Thank you very much. You elegantly showed how chronic stress shifts the approach avoidance balance. Since you're just talking about evolution, how about acute stress, when you immediately have this rush of a danger signal? Have you looked at this, or could you look at this?
Isn't that a wonderful, wonderful question? The answer is no. We're doing a bandit task now in the two-photon work, but it's a great idea. So it's a great idea, and it's just that I hate to tell you how time-consuming this is. We decided not to use virtual reality for particular reasons, namely that it's hard to find place fields in hippocampus, which may have nothing to do with what we find in the striatum, but we're a little worried. But we would love to do that. We are doing, however, a new huge task. We are finding interesting things. It's just that we're in the middle. We don't even fully know the results. I don't hold back anything. Just we're in the middle. But it's a great question. Mickey.
So, your mice in the in the middle seat, who seem to discount avoidance, is that because unpleasant stimuli have in general a lesser effect? They live with them, so they've habituated for unpleasantness, and that might be kind of a sensory input
That's a wonderful
deficit.
Yeah. I completely agree that's a possibility. What we did do, however, was we spent maybe 6 months before we began shaping the animals. And we shaped them so that we could get a reliable percent of approach or avoidance, right? Because we wanted to leave some exploratory going to the bad side, but they were really very highly trained. And so I doubt that that's the issue. In the second set of experiments, the photometry experiments, gee, we've done 15 controls, just everything we could think of, satiety, on and on. So my guess is that's not a major reason, but now I may say that in the monkey, where we revert to the former task, we're looking very, very, very hard at avoidance alone, approach alone, to look at exactly that issue. It's so far, well, it's monkey number one. Yeah. And I've learned from you how to do the monkeys, you guys. Thank you so much.
This is the last.
Oh.
Yeah, quick question. I was wondering about the striosomes. The striatum is a huge structure. So are all the striosomes doing the same thing, or do striosomes in one part of the striatum do something else?
I love that question. So my guess is absolutely not. We concentrated, as I failed, I guess, to say, in the anterior and medial part of the striatum, where these inputs that we know from cortex actually come and we prove it. My concept, so what I haven't told you is that the entire matrix is full of modules. Little finger modules, elbow, shoulder, the whole thing is broken up, somatosensor. We've done a lot of work on that. So imagine an entire, these three-dimensional labyrinths all coming around one another. And so the idea and hypothesis is that striosomes, which are sprinkled through, why are they just sprinkled, right? So one possibility is that they actually have to do with a responsibility signal, as in hierarchical learning algorithms, Quadro and others. And so they can help choose in any given context what should get out. And therefore, I'm almost certain they will do different things. Will they still be, in some sense, evaluative? Maybe yes. Will they do this kind of affect-related evaluation? I'm not sure. But I'll bet you they would have something to do with whether I choose this or I choose that. but I don't know. Great question. All of them are wonderful. Thank you so much.